I'm Jazlyn. I'm currently at the University of Southern California, down the street. But if you haven't met me before, I have come to CGSI since grad school, I think 2017, 2018. I was looking for one of my old CGSI shirts, but all I managed to find was a lanyard from when Eleazar actually sponsored my first SOCNUS attendance. So a student group that I formed here at UCLA as a grad student called Amoeba, it still exists here. We ran a intro to data visualization and stats at SOCNUS, and Eleazar and CGSI sponsored that. So managed to find that lanyard, but not an old t-shirt. So it's weird the things that you find. But today I'll talk to you about some work that I did during my PhD on using dogs as a model system in genomics. I'm hoping I can convince a few of you why this is a great alternate to a biobank. And then I'll talk to you about some recent work that we've done to push the same data forward a bit more. So before I jump into using dogs as a model system, let's see. Let me talk to you a little bit about the origins of dogs themselves. So here what I'm showing in the background is actually from an image from The Origin of Species. So Darwin was actually really into dogs, and he believed that they were hybrids of jackals and wolves, because dogs, one of the interesting reasons why you might want to study them is I believe that they have the largest amount of phenotypic variance of any land mammal. So he was like, there's no way that that dogs originated from one species. They have to be some sort of hybrid. The other interesting thing about dogs is one of the first things that I learned, so this is my dog, you can't give a presentation without dogs and not show off your own dog. So one of the first things that I actually learned is that dogs were the first animal domesticated by man around 15,000 years ago. So just to orient you on the graph, as you get closer to the symbol, you're getting closer to present day time. And as you move from red to blue to gray, you're moving backwards into the past. And so you'll notice up at the top are dogs, and then we have a bunch of livestock below, and cats and chickens all the way at the bottom. But you see this kind of transition from blue to red. And you see the letters W and then the letter D. So what W is these are wild fossil archaeological evidence, so dog-like ancestors. And then D stands for the domesticated version of whatever animal of interest you have. And the different colors just signify remains that we're able to get mitochondrial DNA from versus whole genome ancient DNA sequences. So the blue to red transition is showing you that kind of interaction period where domestication is occurring. So this paper is a little bit old. It's from 2017. We've now found older dog remains, but I think the takeaway here, and the one thing that most researchers will agree on, is that dogs were domesticated around 15,000 years ago. But this is a great article from Holloway. He has a lot of great pieces out there, but I really like this one, which is Dog Genetics Spurs Scientific Spat, especially what's said on the bottom, which is scientists investigating the transformation of wolves into dogs are behaving a bit like the animals they study, as disputes roil among those using genetics to understand dog domestication. So this article basically was summarizing 3 different studies that came out around the same time. And I feel like it was a gloves off kind of article, because one of the authors, this guy Gregor Larson, was basically like, I don't understand why these people are positing that dog origins are where they say they are when they've been on previous papers that say that they're not there and wolves didn't even exist in these regions. So how do we get dog domestication if there were no wolves? So great article if you're interested in learning a bit about the turmoil within dog domestication. So it's been a pretty heated debate for about 30 years, where domestication occurred, how many domestication events occurred, and the timing was under contention for a while, but I think people have pretty much settled around 15,000 years ago now. So just to give you a sense of this, I'll walk through some articles. So here's one of the earlier articles about the origin of dog and domestication. We have some articles using SNP data. We have another one, we're rethinking dog domestication with archaeology and biogeography, so a bunch of different data modalities, looking at village dogs for thinking about dog domestication. using whole genome sequencing now and thinking about dog domestication. This one is suggesting that there's multiple ancient origins of dog domestication. So I think we can all agree to disagree that we don't know where dog domestication occurred. So it's still under debate, and I'll tell you a couple of approaches that people have used because the domestication of dogs is what actually makes them relevant to us in thinking about genomics. So what I'm showing here is one of the first papers that I read when I transitioned and started thinking about dogs for genomics. So it's a little bit busy, but I'll walk you through it. So what that gray area in the plot is showing in this figure is the range of wolves before dogs were domesticated. So that's wolf range. And then each of the green circles is representing archaeological remains. If the circle is filled in, that's the oldest remains that we had at this time. So this is from 2012. Now there's even older remains. And then as that pie is getting more and more white space, that means the remains are younger and younger. And then what you see next to these pies and in regions that I've circled are little kind of icons of dogs. So these dogs are kind of these weird basal breeds of dogs that stand out and look quite differentiated from the classic breed dogs like the collies or the retrievers that we see today. So these are breeds that are like Basenjis, Akitas, ancient Spitz groups, Mastiffs are in this. And what the authors in here argue is that, so some of these regions that I've highlighted are the most I should say the most frequently used for dog domestication events. So they argue that Europe and the Middle East have the oldest archaeological evidence of dogs, so domestication probably occurred there. But then you get people who are coming back and saying, are we sure that these remains are actually dogs? Maybe they're more wolf-like. So ancient DNA can't tell us the full story. Some people have also advocated that potentially dogs originated in Central Asia, in Siberia, and even in East Asia because the mitochondrial haplotypes in East Asia looked the most diverse. So there's no agreement here. But the thing, or the article that convinced me of the origins of domestic dog was actually a paper that came out in PNAS. So this is what's called a village dog on the cover image. This village dog is in, I think it's Nepal that this picture was taken. And have people heard of village dogs before? No? So it turns out that village dogs are actually the most abundant dog. So they're the transition between, so that wolves were domesticated, we get wild village dogs. They're free-ranging, free-roaming. They're the most abundant dogs, so they're domesticated. They live around the world. And then these village dogs went through the breed formation bottleneck, and that's where we get the collies, the labs, the retrievers that we see today. So they're kind of this intermediate point between dogs and wolves. And like I mentioned, village dogs exist around the world. So there was this village dog genome project that happened. So what authors did was go take samples from over 500 village dogs from more than 40 countries around the world. And what I'm showing here in PC space next to the picture of the cute village dogs is their genetic diversity. So the black dots that are filled in are these classic kind of European breed dogs, and then the ones that aren't filled in are these more basal dog breeds like the Chow Chow, the Akita, the Basenji. But what you see is that the village dogs have a lot more genetic diversity than do the breed dogs, but they also share a lot of their genetic diversity. And there's these really interesting stories about dogs that are actually on these islands like French Polynesia and Fiji. So really cool backstories there about how a lot of them are actually more European than you would expect. So we know they have a lot of diversity, and maybe rather than thinking about archaeological remains, we can think about this intermediate village dog for discovering where domestication occurred. And the author of this paper is a guy named Adam Boyko. It's coming from Adam Boyko's group, one of his students, and he actually has training in human genetics as well. So he kind of took the perspective of human geneticists for trying to figure out where were dogs domesticated. And I think that a lot of it came from this really nice paper from Matthias Jacobsen in 2008. It's one of the first papers that I read. But the idea is this. So behind me, what I'm showing you is a map of the human migration out of Africa. Also contended where we originated in Africa, so this is just one possible version, but I think what we can agree on is somewhere between 50 and 60,000 years ago, humans began to migrate out of Africa and people the rest of the world. And so what the arrows are showing are migration routes throughout the world, and then the thicker arrow is what's called a founder event, meaning that we had a population, and then when migration occurred, a new population split off of that original group. So that new population is smaller than the original group, and it also has a subset of the diversity of the original group. And let's see if I can put a pointer on here. Oh, maybe it's not gonna show up. Oh, there it is. Okay, sweet. So what this is showing over here is this concept of linkage disequilibrium. We use it a lot, right, in human genetics. Linkage disequilibrium is just the non-random association of alleles of different loci. We use it for imputation, we use it for GWAS, we use it for inferring demography. But the idea is that these populations, these African populations that have not gone through this series of bottlenecks during migration have a much faster rate of linkage decay, and two, they have lower linkage disequilibrium than these American populations that have experienced many subsequent bottlenecks as we moved throughout the world. So another way to think about that is just that as your distance from East Africa increases, we expect to see more LD in these populations. So he thought, can we basically do this in dogs and try to figure out where domestication occurred? So we know that, okay, that dogs, horrible schematic, but it gets the job done. At some point in time, there were gray wolves. They were domesticated. We see village dogs, and then there was a breed formation bottleneck, and that's how we get the classic breeds that we see today. So work previous to his had looked at LD within dogs. So the solid lines are real data. The circles are just simulations that this study did. And what I want to highlight is that here's wolf LD decay. It looks a lot like we expect to see in African populations, right? Wolves have not experienced bottlenecks. They have lower LD. And then we see this kind of range in how high LD is in different breeds of dogs. So each line is a different breed of dog. And we can see that it varies quite a bit. And that's actually a result of the fact that what I'm showing here, so these blue arrows represent where the domestication bottleneck starts. Each of these is a group of dogs, a breed of dog. And then on the y-axis is the effective population size. I wouldn't pay so much attention to that as just the trajectory. What we find is that there's different numbers of sires and dams that founded breeds. So basically what's happening is the strength of your bottleneck varies, and that's what's responsible for these varying levels of LD in dogs. So that's great that we see varying LD in dogs. Oh, let's see if I can turn this off. But that doesn't actually answer the question of where domestication occurred, because breed dogs have all gone through this breed formation bottleneck. So if we want to figure out where domestication occurred, we want to focus on these village dogs. And the idea here is that they're free-ranging, free-breeding. They shouldn't have experienced a bottleneck other than basically when they first arrived at the area that they're in. And that's also what we see in data. So I'm pointing to the same place where the breed formation bottleneck occurs. And what we find in village dogs and wolves is that their population size is pretty much stable. So we don't observe that bottleneck at all. So when we go and we look at LD, what we find is that wolves are right here. And then here's different village dogs that were sampled around the world. And this is a key feature, and this is what makes studying dogs quite powerful, is this linkage. And if you zoom in on basically super short distances, what we find is that this curve here, which represents Central Asia, no matter the distance that you're looking at, within a short range, it always has the lowest amount of LD and the fastest decay, so it's the closest to wolves. So the authors here advocate that perhaps this means that domestication occurred once in Central Asia. They do some additional modeling to show this, and that the common ancestor of all breed dogs is actually in Central Asia. So I fully bought that. I thought, you know, for sure everyone's going to agree, but it turns out no. In 2020, another paper came out with ancient DNA and said that's completely wrong. If you look at archaeological remains, domestication actually occurred in the Middle East and North Africa. So there's still no agreement. And I think that this is a really great area of dogs to study. But maybe the answer is that we'll just never agree on it. But if you want to move into aDNA, this is a place to go. It will certainly get you a lot of science and PNAS papers. So I think what I can conclude with is the origins of dogs are unknown, or at least their ancient origins are. Super contentious, but it's actually their very recent history that's our friend and what will help us. So what I mean by recent history is this breed formation period. So what I'm highlighting in red. So what this breed formation period has done is that it has simplified complex trait architecture in dogs. And I'll show you an example that I hope will win you over for advocating how this simplification has occurred. And I'll use a very classically studied trait for this example. So here I'm showing, I think it's called Joyner's living histogram of human height. And then what I'm showing you is a range of height in dogs. So we have the smallest dog, the little chihuahuas, right? And then the largest dogs over here, they measure them from like the foot to the withers, which is their shoulder, is this Irish wolfhound. So there's a huge range of variance in the size of dogs. We don't see that much in humans. And this is actually one of the largest GWASs that was done on dogs. Any guesses for how many dogs were included in this GWAS? Thoughts? How many? 420. Okay, 400. Any other guesses? What would you need for a human study to get p-values like this in your Manhattan plot? Maybe a little more, like 1,000, right? Maybe more than that. Okay, 50,000, we're good. Okay, 2,000. So 2,000 breed dogs representing 158 breeds. This is just looking at males in this graphic, produced this Manhattan plot. And so just to get a sense of this, I just ripped a random paper off the internet. But this is, on the bottom, a GWAS in height in 10,000 Europeans. P-value is nowhere near the scale that we're seeing in dogs. So we're not even getting close to what we're seeing in just under 2,000 dogs. And in fact, I think this came out pretty recently. I'm not as up to date on the human genetics stuff as I should be, at least with height. But this is one of the largest studies that was done. It's a meta-analysis of over 280 studies of humans, over 5 million humans. And we are finding like 12,000 SNPs that are associated with human height. In contrast to the 17 loci in dogs that actually explain the vast majority of the variance in height. So in green is height down here, and what you find is that these 17 loci are explaining over 80% of the variance in dog height, and when you add this correction factor for the breed of the dog, they explain almost all of it. So I needed less than 2,000 dogs to get me to the scale of what 5 million humans isn't even providing me, because those 5 million humans, it turns out, we're explaining like 50% of the variance in height. This is just common SNPs of Europeans. Even less when you look at the other populations, so you'll notice different colored dots there. Even less when we look at these other populations. So that's crazy. Definitely a reason to study dogs. We can see that there are actually very few loci that are associated with most of the traits, and this is actually still one of the largest GWASs that's been done with dogs. And the reason why that is because of their recent breed formation bottleneck, the recent history. So what it's basically done is For most breed dogs, what we find is that variation within them is fixed and shared among all the individuals. And since most of the variation in there is fixed, the very few SNPs that occur have super large effects. This is in contrast to humans, where we find these tiny, tiny effect sizes. In dogs, the few SNPs that they have very, very large effect, and we have very large extended blocks of LD. So one thing that we do have to tackle in dogs, though, is that fine mapping is hard to do. So in humans, LD decays much faster. Fine mapping's a bit easier to do. In dogs, it's super hard to do fine mapping. So that is one kind of point that makes it a little bit difficult. But like I mentioned, because a lot of the alleles are homozygous due to this breed formation bottleneck, the concept that I've always been interested in is thinking about runs of homozygosity and identity by descent. What that means is that breed formation has also increased the amount of identity by descent segments and runs of homozygosity in dogs as well. And I'll walk through what those are just so we're all on the same page. So here what I'm showing behind me is a pedigree. And what identity by descent actually is just when two people or two organisms inherit the same piece of chromosome from a common ancestor. So here that might be the light orange segment. You can trace it throughout the pedigree, and we see that these two first cousins have inherited the same light orange segment, IBD, from their grandparent. And then all runs of homozygosity is just a special case of IBD that's a result of inbreeding. And so dogs are super inbred. There are actually very few individuals that founded any given breed. Some breeds have almost gone to extinction and then were rescued by like 2 individuals. So they're super inbred. And what that does, what inbreeding does, is it converts these light orange segments that might be IBD within a large population into runs of homozygosity. So dog demography has increased runs of homozygosity, and it's these runs of homozygosity that I'm interested in because I think we think a lot about genetics in kind of this additive framework. I'm more interested in it from the standpoint of recessive variation, and at least for dogs, I think the major conclusion is whatever can become homozygous and you're gonna make it through has probably become homozygous in the dog. So for example, when you think of pugs, right, they have the super smushed face. If I released a pug in the wild, it's probably not gonna make it. But because it lives with me at home, it's gonna make it. So I kind of think of it in the case of like, you know, if it can make it, we're providing all the conditions for it to survive. Because we've applied such strong artificial selection, basically everything is homozygous. There's very few SNPs that are there. What is there is like actually working the way that we wanted GWAS to work, right? Originally it was kind of this common disease, common variant. The SNPs are common and they're at an appreciable frequency within the breed. So the question is, that I wanted to approach during my PhD is, can we detect the association between runs of homozygosity and complex traits? And the best place to start is probably dogs. So what I did was I aggregated a bunch of data, array data from 4,300 dogs and wolves. They represented around 200 breeds of dogs and then 5 populations of wolves. And I had case control status for a subset of this data. Originally, I just focused on these kind of 12 clinical and morphological phenotypes. And then more recently, we've expanded that. And basically, all I want to do is test whether the burden or the total amount of the genome within a run in a dog is associated with any of these clinical or morphological phenotypes. But in order to do this, we run into a very common theme that we also see in human height. So if people have been around, there was a whole lot, there were a couple papers that came out about the Giant Consortium and UK Biobank and how we actually have to be very careful about how we correct for structure, even in European populations. In dogs, the case is even worse. So here what I'm doing is I'm showing, and maybe actually I hope this shows up a little bit better, is the distribution. So F row is just taking the total amount of an individual's genome that's in a run, dividing by the total length of the genome. And here what I've done is I've grouped dogs into basically their larger clade. What you can see is that there's a lot of variation in the total amount of the genome that's within runs across these clades. Some of them have insanely high inbreeding coefficients. And we also see that dogs in general have higher inbreeding and more runs than do wolves. This makes sense. Most wolves haven't gone through a giant bottleneck. Some of them actually have, so like this Italian wolves in the US have gone to near extinction, and they've had to be genetically rescued by very few individuals. There's also a population in Isle Royale where the same thing has occurred. So we have to be very careful about how we account for structure, but we can do it. And because we're looking at runs, we are doing less tests than if we look at SNPs. So we also have power here as far as like doing the multiple test correction. But how do we account for structure? So one way, and I hijacked this, there's a lot of great papers in plants from thinking about plants, is we could just correct for breed structure by looking at pairwise sharing of ROH. So let's take individual 2 as our kind of focal representative individual, and then each of these lines is representing a run of homozygosity. So all I'm doing to create our relatedness matrix is going along the genome and asking what's the overlap between individual 2, individual 1, individual 2, individual 3, and I repeat that for my whole data set. So here it might be 5 base pairs and 2 base pairs. I slide along to a new region of the genome. There might be no overlap. And then what I can do is create my relatedness matrix that's based on ROH sharing. And this is because I want to test the burden of runs of homozygosity or inbreeding in a trait, not going SNP by SNP. So this relatedness matrix is not that bad to create. I also tested just using a traditional relatedness matrix based on SNPs and PCs, saw very similar answers. This matrix also recovers breed structure, so it's doing the things that it needs to do. So because it's working, it was firing on all cylinders, and I was trying to get out of my PhD, I could finally ask the question, is there a connection with genomic sharing in the form of runs of homozygosity or inbreeding and disease? So just to make sure that that's just clear in your head, so the idea is something like this in this flowchart. So we have these rare deleterious mutations that are occurring somewhere in the lineage before breed formation. And then during breed formation, we have tons of inbreeding. We get our boatload of runs of homozygosity, and these mutations that are recessive are unmasked. They're now homozygous in our dogs. But let's imagine these mutations aren't lethal, right? So that means that the offspring are viable, but they will have some sort of disease. And so what we would expect is some enrichment of runs of homozygosity in regions with these recessive mutations. We might expect this. And we also expected that there should be some association between runs and disease. So the next question is, do we actually see that? So what I'm showing you above, so remember on the x-axis, this is actually a beta that's estimating the log odds change in risk for a 1 unit change in ROH, because I'm looking at disease traits here. As we move to the right, we're seeing ROH increasing our risk. The other side, maybe the runs are protective, and this is also super interesting too. But what we saw was that there's more significant associations, or nominally significant associations, than we expected by chance. And I want to focus in on the super interesting run, which is one where we had the most data, which is lymphoma in golden retrievers. And before I jump into that, I just want to highlight that right above it is lymphoma in Labrador retrievers, and you notice that it's going the opposite direction. And then when we aggregate the data together, we see no signal at all. So I thought that was super interesting. But maybe the most interesting, and the one that we had the most data for further exploring, was this lymphoma in golden retrievers. So the question is recessive variation that's in these runs of homozygosity potentially associated with lymphoma? That'd be super cool. So the first thing to note is that the sample sizes are actually really, really small. So what I did was I just plotted how much of the genome is within runs and cases and controls. And what we saw is that cases on average carried more of their genome within a run than controls did. And the second thing that I did to try to get a little bit closer to answering this question was I asked how many of those runs are actually overlapping genes associated with lymphoma. The hard part is there's not a lot of studies that have been done in dogs, but there are more that have been done in humans. So what I did is I aggregated genes that have been associated with lymphoma in humans and dogs, and then used a permutation test to ask whether more runs overlap these lymphoma-associated genes. So it wasn't significant, but it looks like maybe if we can get a lot more data, there's a chance there. Could be super interesting to explore. So I thought this was super cool. More recently, we went on to test binary phenotypes. And what we found there is, once again, we have these cases where ROH seemed to increase these like white chest, white head, muscled. There's been more work that's shown that there are homozygous variants that are associated with giving you these phenotypic outcomes. We have cases where ROH are decreasing it. And then the more important one is maybe continuous traits. So this is work that was actually led by a super talented undergrad. And so what she did is she went back and she looked at continuous traits that we also had data for. And maybe the most interesting ones and the ones that stood out to me were lifespan and height, because I've been trying to make this case for studying dogs by using height. And lifespan is one where this was actually a shout out to Lisa. She said in our biobank session that, you know, we always can get new data. We can always get larger biobanks or move in QCR data sets. But the real question is, do your results make sense? Thinking back to Luz's talk yesterday, like, what about ascertainment bias? So one thing that you want to always do when you're using these large data sets is say, like, is there a result that I expect, and am I actually getting that result back? So previous work in dogs, and this is just using pedigrees and super small sample sizes, what it had shown was that individuals who were inbred lived, their lifespan was decreased versus those that were outbred. And that's the exact same result that we saw for lifespan, which is more on the order, though, of like F-row being associated with the trait, but like F-row in all individuals, what it seems to do is decrease the length of life. And the other thing that we also saw is when we go to height and we zoom in a little bit more, and so here what I'm showing you are Manhattan plots where we're looking at runs and we're associating the actual run itself with the trait. On the top, we're doing this association test with all of the dogs. In the middle is small dogs. And on the bottom are large dogs. So then we've mapped, you know, or labeled which loci are showing up as hits in this association test with heights. Overall, what we found is that the vast majority of genes that were identified as being in a run and associated with this trait have been previously identified in GWAS for height in humans. So most of them have been hits, and the real kind of confirmation that we had that this is working, this is giving us very, very sensible results, was actually looking at IGF. So there was this cool science paper, actually coming from, it's this one here, the Sutter 2007 paper. On the cover was like a giant Great Dane looking down at a Chihuahua. And what they said is this gene, IGF-1, is actually explains, I think, almost like 70% of the variance in height between a Great Dane and a Chihuahua. And in small dogs, which are shown in red, and in large dogs, which are shown in blue, in small dogs, what we saw, they actually went in and did fine mapping, is that at this position, most small dogs were homozygous, whereas large dogs were kind of all over the place. Sometimes they were homozygous for the other allele, sometimes they were hets, mixed breeds kind of fell in the middle too. So if this result is true, right, what we should see is that there's some association with a run of homozygosity and height in dogs, and that's actually what we do see in our data as well. So this is telling us, like, these are very sensible results. It makes sense. There's some recessive variation that is responsible for height and a number of other traits in dogs, and we can do these tests with very, very few dogs, many less than we need for humans. So they're a great alternate to a biobank. And perhaps I haven't convinced you enough of that, but there's actually a lot of common diseases that are shared between humans and dogs, and there's lots of room for doing translational work. So actually, narcolepsy, or a gene that was implicated in narcolepsy, was originally discovered in Doberman pinschers in labs. And then, so this is 1999, and then very soon after, human geneticists went and looked in humans, and they found that I think something like 20% of the cases where the individuals had narcolepsy and they couldn't figure out why, where actually it was the exact mutation in the exact same gene. So this is not a rare occurrence. There's lots of room for translating results that we get in dogs to humans. So super cool. There's also a great model for muscular dystrophy too. So good research that's going on in the dog world. Great work on aging as well, and cancer. And there's room to work in that space. And the last thing that I want to end with is just a cautionary tale. So this came from asking the question, is the prevalence of ROH, or identity by descent segments, associated with the incidence of recessive genetic disease in any dog breed? So this was inspired a lot by human genetics work, where we saw that basically as runs of homozygosity increase, so these are just different classes of runs here, we see more damaging deleterious mutations accruing within them. And then a more recent paper in dogs showed that as the length of runs increases, you're seeing at a minimum a 30 times higher inflation of deleterious variation. So they're capturing more and more of those deleterious variants. So the question is, right, as the amount of the genome in a run increases, we should see more recessive disease-associated variants. We're increasing inbreeding, we're increasing disease, so that as we get to the most inbred dogs, right, they should be the most diseased. They're 2 heads, 2 tails, they should be doing real bad. But how do we actually quantify this? We can just compute a breed score of runs, or IBD. So all you're doing is summing up the total length of IBD or ROHs, and then you're normalizing for all the comparisons you're doing or all the individuals that you have. So most diseased dogs should have the largest amount of recessive variation in them. So I do this, I make my first plot, and I get this result. So I was mad. I was like, was I wrong? This is the exact opposite expectation. So those breeds with the largest amount of IBD have no causal variants that have been identified in them. And this is coming from the animal version of OMIM. It's called OMIA. So there's 0 causal variants for most of these things, or 1. All the ones that have the majority of the causal variants are floating somewhere over here, and they have the lowest amount of IBD. So I was like, you know what? Maybe IBD was the wrong approach. What about just looking at runs? Same thing. And I was like, how? This doesn't make sense. And then I'm not big on dogs, so I had to say well, what exactly is this thing? I don't know what a bearded collie is. I have no clue what a New Guinea singing dog is. What's a Bedlington Terrier? What are these things? So I'm looking them up online, and then it dawned on me, these can't be very common. But you know what I definitely know is what a lab is. I can immediately think of that. I can definitely tell you what a German Shepherd is, Golden Retriever, Beagle, Dachshund. So what the AKC does is they keep records of how popular a breed is through time. And if we basically just take the integrals to the area under the curve of this popularity through time, or how many individuals, we use basically how many individuals are registered in the breed, what we suddenly find is this positive correlation between how popular the breed actually is and the number of causal variants being identified. So this is the exact same ascertainment bias that we see in human data. There's a great paper from Molly Praswarski's group with Eduardo Amorim, where they show this ascertainment bias in Finnish data. Same thing that we see in dogs. So cautionary tale, we do still see the same biases in dog data, where it's probably the popularity of the breed that's driving discovery, and also the money that these breed groups have. So a lot of the studies that are on hey, my Golden Retriever, I'm noticing it's dislocating its shoulder a lot. It's funded by a breed group. So there's biases there. So in closing, I hope I've advocated for the fact that we can use dogs as a model organism to study disease. Dog domestication, don't try to get into an argument about it unless you're really ready to get into an argument. It's complicated. But what is super cool is that breed formation has simplified complex traits. I hope I've shown you that runs of homozygosity can be connected to disease status for some breeds. We see the same thing in human populations as well with many more samples. And we can use runs to identify regions of the genome that are associated with numerous traits. But these runs are also linked to inbreeding depression. And lastly, give yourself some caution. Always test your data. Much like human data, there are biases in dog data as well. And a lot of things might actually be linked to how many times the dog is, or how prevalent is the dog, how often is it being seen by vets, more so than it's being led by biology. So with that, I hope we have a little bit of time for questions, but thanks to my PhD lab, Kirk, and Abby, who was a talented undergrad who led some of the analyses, and then more recent work with Zach and Sweetlana. And I can take maybe one question or two. Thank you.